Okay, right. Good morning. Um, so we're going to continue talking about Mendel's laws. And where we left off, Mendel's work had fallen into obscurity for 35 years, and then it got rediscovered in 1900. And it was rapidly realized that the laws that he'd worked out applied to a lot of species, not just peas. And in fact, they apply to cats as well, like this annoying beast right here, who, as you can see, is a black cat with some white on him. He's got some white on his face. Oh, that's very sweet, kitty. And white feet, and yeah, white hind feet, and white front paws, and white on his face right there. And I'm going to set you down right now. Good kitty. And we can use what we know about Mendel's laws to explain why that cat looks the way he does. But there's a few more principles that we've got to add to it. We've got to extend Mendel's laws a little bit, as people learned how to do when they were rediscovered in the early 20th century. So without further ado, let's get on over to slides. And here we are, or should be. And so we'll start with the rediscovery. Up. Okay, there. Okay, sorry, the system's taking a little time to. All you have to do is, you don't have to know, for purposes of this class, the basis is for why cats are black versus brown. I mean, unless you really want to, or unless you become a cat breeder or something like that. What you do need to know is the principles that the case studies illustrate. I'll never ask you a question on cats on the exam, but I might ask you a question about reticulated pythons or something like that. And you just might have, and I won't have lectured about reticulated pythons, but you will have to know the genetics principles. So I'm going to see if I need to let somebody else in. And I'm sorry, my system is kind of full right now, but here we go. So I hear somebody knocking. Okay, sorry, I'm waiting for the ding ding. Okay, good morning. And right, let's get back to all right, let's get back to the slides. Okay, here's the first thing. With the examples that we looked at of Mendel's peels, there was purple or white flower color. However, we now know that alleles don't come in only two forms. And one example is that there are actually 3 alleles of the gene for a dark base color in cats. We can write these capital B, lowercase b, and lowercase b with a tick mark next to it, which we pronounced B prime. And in this case, they form what we call a dominance hierarchy, in that black, which we write capital B, is dominant to medium brown and it's dominant to light brown. So a cat that is big B, big B, big B, little b, or big B, little b prime will be black. Medium brown is recessive to black, but it's dominant to light brown. So medium brown, we write little b. A cat that is a medium brown color will have the genotype little b, or little b prime. And then light brown is recessive to everything. The only genotype of a light brown cat would be little b prime, little b prime. And breeders refer to these, by the way, as chocolate and cinnamon. And there's what they look like. There's a black cat up at the top left, which could have any one of 3 different genotypes. There's a dark brown cat at the lower left that can have any one of 2 genotypes, and then there's only one way to get a light brown cat to be homozygous for the light brown allele, or should I say the light brown allele. Right, sorry about that. This matters for something that you have to do in class, because the lab for this week, which I uploaded to the usual spot yesterday, involves you knowing something about human blood groups. This was discovered by a guy named Carl Landsteiner, who won, I believe, the very first Nobel Prize for medicine because it made blood transfusion safe. And it's saved countless lives since then. There's actually a number of human blood groups. ABO is probably the best known. You probably also know about the RH blood group, how you can be RH positive or RH negative. There's actually others that aren't usually that crucial for medicine, except when you're doing organ transplants or something like that. But the ABO blood group is controlled by a single gene with 3 alleles. We can abbreviate those as A, B, and O, although the pros will sometimes use capital I with an A superscript, right? That's a capital I with a small capital A above and to the right of it for the A allele, and capital I superscript B for the B allele, and then lowercase I for the O allele. How you choose to write it, I'm not going to worry about. But the A and the B alleles are co-dominant to the O allele. If you've got type AB blood, then you've got one copy of the A allele from one parent and one copy of the B allele from the other parent. Doesn't matter whether you got A from your mom and B from your dad or vice versa. If you have type A blood, you could have 2 copies of the A allele, one from mom and one from dad, or an A and an O, one from the A from one parent and the O from the other. If you've got type B blood, you could have genotype BB, or you could have BO. And if you have type O blood, the only way you can have that is if you have the genotype OO. This has been used in paternity testing, by the way. You can't use it to prove that so-and-so is, the father of such and such, but you can rule it out. You can sometimes rule people out as the father. So, for example, I happen to have type O blood, so my genotype must be OO. There is no way that I could ever have a child with type AB blood. because someone with AB blood must have gotten either an A allele or a B allele from the child's father, and I don't have an A or a B allele to give because my genotype has to be OO. So if somebody were to sue me for child support and the baby turned out to have type AB blood, there's no biological way I could be the father. And that's been used in cases before. There's a lot more sophisticated genetic testing that you can use these days, but you'll actually work with this in actually in the lab that I uploaded for you to do yesterday. There's another phenomenon called incomplete dominance, and I'll first show it to you in some flowers, not peas, but snapdragons in this case. And it turns out that if you cross a red snapdragon with a white snapdragon, the offspring are all pink snapdragons. That seems like it violates what we covered earlier. That seems like it violates the principle that of dominance, right? When we crossed a purple flowered pea with a white flowered pea, we didn't get lavender peas or pink peas or anything like that. Turns out that if you cross two pink snapdragons, you get one red and one white for every two pinks. And the reason why that's the case is that the red allele, which we can write capital R, does not completely hide the white allele. We call that incomplete dominance. The purple flower color in peas does completely mask the presence of the white allele. The red allele in snapdragons doesn't completely mask the white allele. So heterozygotes are pink, and if we cross two snapdragons that have the genotype big R, little r, then it follows your basic Punnett square rules, but we get one red, big R, for every two pink, which are heterozygotes, big R, little r, for every one white, little r, little r. So that follows the exact same rules as pea plant flower color, except for the fact that in this case, we can tell the difference between homozygotes and heterozygotes because the dominant allele does not completely hide the recessive allele. And if you want a cool example of this in kitty cats, there's an allele for white spotting, and it happens to be dominant to the allele for solid color. But it's incompletely dominant. So on the left, that is a black cat, but that's a cat that is homozygous for the dominant allele for white spotting. And it's basically a black cat with so many white spots on its body that it's mostly a white cat. Pretty much the only black that you've got left is going to be around the ears. On the right, you've got a black cat with the homozygous recessive genotype, and that cat is solid black all the way through. That cat, by the way, is also one of ours. That's Boudreaux over there on the right. And in the middle, that's a black cat that's heterozygous. And you can see that cat has white spotting, but not as much as the cat on the left. So-called tuxedo cats, like Bootstrap, the one I was wrestling when we started here. Those cats are heterozygous for the white spotting allele, and they might only have a little, white star on the chest or white socks or, a white face to some degree. But they're kind of in between. So that white spotting gene does not completely hide the presence of the other allele. Okay, I'm assuming I'm making sense. Okay. We also have to get into a phenomenon that's very, very common called epistasis. And what that means is you have several genes that affect the same trait. And you can have one gene with alleles that modify the effect of another gene. So this cat right here, breeders would call this blue. This may be what they call a Russian blue cat, although it looks to me more gray than blue. This is a black cat, so this cat might have genotype big B, you know, like a regular old black cat, but this cat is also homozygous for an allele that takes whatever the cat's base color is and makes it look lighter. It's called the dilute allele. We can write it as lowercase d, and it's recessive to the non-dilute allele called dense, which is big D. And the dense and dilute alleles don't control what color the cat is. They're not genes for gray fur. They are genes for whatever the cat's color is, lighten it up a notch by affecting the way that the pigment is deployed in the hairs when they're forming. So on the left, you've got a cat that is genetically black. On the right, I don't know why I have top and bottom there. That's an error. But you've got a black cat on the left and a medium brown or chocolate brown cat on the right. But both of them also have the genotype little d, which says whatever the base color is, lighten it by several notches. So they've got the dilute black, which looks gray, and dilute medium brown, which looks tan. For some reason, cat breeders like to call these colors blue and lilac. I'm perfectly happy with gray and tan, but you know, nobody asked me. Okay, you with me there? So that's a simple example of epistasis, which is what you get when you have one gene affecting the expression of another gene. There's no gene for gray fur color. There's a gene for black pigment, and there's a gene for take whatever the base color is and lighten it. And this sort of thing actually is extremely widespread. And here's an example. I sometimes get asked about human skin color, and the answer is that last I looked, and this was a couple of years ago, there were no fewer than 9 different genes that had a strong effect on skin color. There are more than that that can, you know, tweak the tone a little bit. But there are 9 genes, each of which has 2 or more alleles that have a noticeable effect on how much brown pigment you put into human skin. This means that Punnett squares on human skin color would be extremely difficult to do because you'd have to have something like a 2048 by 2048 Punnett square. And this is why human skin color does not really fall into neat classes. I mean, we talk about, you know, Michael Jackson said, you know, it don't matter if you're black or white, and you know, Paul McCartney talked about ebony and ivory, but most of us are actually kind of somewhere in between. You know, we're all varying shades of, you know, brownish peach, more or less. And that's because there are so many possible genotypes, you know, where you have to take into account 9 different genes, each of which has at least 2 alleles, that human skin color is basically a continuous range. We don't really fall into black or white categories in the way that, you know, pea plants can always be grouped as either purple flowers or flowers. With us, there's an awful lot of in between, and it ultimately results from epistasis, from the fact that there is no single gene that determines your skin color. There's at least 9 major ones that have an effect on skin color. And so the genotypes can get very complex indeed. Okay, so that's epistasis, 2 or more genes affecting one trait. The, I guess, opposite would be pleiotropy, which is a case where one gene does multiple things to the phenotype. So you can have one gene that has multiple effects in pleiotropy. And in epistasis, you have many genes that all contribute to one trait. And so here's my cat example for you. This is a breed of cat known as the Devon Rex. Rex cats are called that because they've got wavy fur. I'm not really sure why they picked the word Rex, but there it is. A Rex cat is a cat with wavy fur. Most cats that you'll see have very straight fur. And the so-called Devon Rex allele is recessive to the non-Devon Rex allele. Just about all the cats that you've ever seen would just be homozygous for the non-Devon allele. And if you have 2 copies of the Devon Rex allele, it gives you wavy fur, but it also gives a cat huge ears that are set low and a wide face. Devon Rex cats sometimes are said to look like Yoda. So here you've got some Devon Rex kittens. I'm just imagining you all going, "Aww." And you can see they've got great big ears and this very wide face with the low set ears that makes them look like Yoda. Or, you know, sometimes I've heard them called fairy cats or pixie cats or something like that. And so there you have one gene that has 3 different effects on phenotype: wavy hair and big low set ears and that wide pixie face. It's cute. Right. Cute they are. Somewhat sadder example of pleiotropy. There's several ways you can genetically get a white cat. There are some cats that are called white when they're basically homozygous for that white spotted allele, and they've got so many white spots that it basically covers up the color. But there's other genetics ways that you can get a white cat. And cats that are homozygous for one white fur allele also have blue eyes, and also they're usually deaf. There's actually kind of a neat linkage between the production of pigment in the skin and the development of hair cells in the inner ear that allow you to perceive sound. Don't really have time to get into that, I'm afraid. But yeah, cats that are homozygous for one of the white fur alleles also have blue eyes and also are usually deaf. Fortunately, the 2 white cats that we've got at our house are not deaf. I'm not sure of the details. I can't even really do a Punnett square because, you know, I don't know where they came from. They were just strays. But genetically, they've got a different background, a different reason for having white fur. So they can hear just fine. They just ignore me. Right. If you want a dog example, because we've got at least one dog owner in this class, there is a coat pattern in many breeds is called Merle. I had a dachshund when I was a kid who had this coat pattern, and in the case of dachshunds, for some reason they call it BIOL. In others, it's called Merle. It's the same thing. It's the same, you know, genetic basis. And Merle, what you have is a coat pattern where there's big patches of whatever the base color is are lightened. So here you've got a dog with black patches and gray patches, and blue eyes are very typical of Merle dogs. And this is typical in some breeds. Now, it turns out that Merle is dominant to non-Merle. But if you have a dog that is homozygous for Merle, so genotype big M, they lack pigment almost completely, and they also develop abnormal eyes and ears, so they're usually blind and deaf. This very unfortunate dog is a double merle. And this is why you should never breed two merles together, because if you do, roughly 25% of your offspring are going to be double merle and not only lack pigment, but they'll be severely handicapped with inability to see or hear. Something to keep in mind if you ever do dog breeding. It's recommended that you not breed two merles. This healthy merle right here must be heterozygous, genotype big M, little m. And it can get worse. This is a breed of cat called a Manx cat. Manxes have no tails, and they're born that way. They're not docked or anything. They're born tailless. They tend to have very long hind legs, and without that tail to balance them, they don't run so much as kind of hop. So I've actually heard them called cabots, not because they come from a suburb of North Little Rock, but because people used to think they were crosses between cats and rabbits. That can't actually happen, but it's a consequence of having no tail, is they tend to run funny. Now, this is caused by a single gene with two alleles. Tailless, big T, is dominant to the tailed allele. The problem is if you crossbreed two Manx cats, you'll get two tailless kittens for every tailed kitten. And the reason is that the genotype big T is embryonic lethal, meaning it causes the embryo to fail to develop. A fertilized cat egg with the big T genotype will never be born. The embryo will stop developing and essentially be miscarried. So any Manx cat that you see must have the genotype big T, little t, right? That has to be the genotype of that cat right there. And crossing two Manx cats is crossing big T, little t with big T, little t, and that will give you one big T cat, which is never born, and two big T, little t Manx cats, and one little t cat, which will have a tail. So that's why you get this 2-to-1 ratio instead of a 3-to-1, which you might expect. The reason is that some of your offspring are never seen because they just don't develop beyond a very small embryonic stage. So yeah, you can have Mendelian ratios distorted if some of the possible genotypes are embryonic lethals, incompatible with life. Poor kitties. Right. Last thing I'll talk about before the break. Genes are also influenced in a lot of ways, many of which we just don't have time to get into, by the environment that they're in. Genes are not just magically doing these things in a vacuum. There are lots of ways in which the environment can affect gene expression, and there's a beautiful example of this in kitty cats. There's an allele for color, and it's written capital C, and a cat that has at least one copy of the color allele will basically have color. This is another example of epistasis. A black cat will have, genotype big B, but a black cat must also have, at least one dominant big C allele in order to make any pigment at all. So this is another case of epistasis. You've got to have at least one color allele present for the cat to show black, brown, orange, gray, lilac, cinnamon, whatever it is. And that color allele is dominant to the albino allele, which is little c. A cat that is homozygous for the albino allele will be completely white with pink eyes. Not very common because I think albino cats are not real healthy. But remember that many genes have more than two alleles. And there's another allele of this gene called little c superscript s. And a cat that is homozygous for that will express whatever its base color is: black, brown, orange, tabby stripes, what have you. But the color will only be expressed on the parts of the cat that are cooler. The warm parts of the cat will not express that color, and they will look white. So think about this for a minute. The parts of a cat's body that are cool are pretty much the same parts that get cold on you when you go out underprotected on a winter day, which actually sounds really nice right about now. Love to be able to stand outside on a winter day, especially yesterday. So if you think about it, if you're outside on a cold day, your face gets cold, your ears get cold, and your hands and feet will get cold. Here we've got cats that are homozygous for that CS allele, and they're showing pigment on the same places. The face, the ears, you can't see it in this picture, but the paws and the tip of the tail. And these are, of course, Siamese pattern cats. So what you've got on the left is a black cat. That cat might well be homozygous for that big B allele, but it's also homozygous for an allele that only puts down color in the parts of the body that get cool. On the right, you've got a cat that is homozygous for the Siamese pattern. It's got a light brown color, so its genotype somewhere, if you go back, is like little b prime, but it can only show that color on parts of the body that get cool. In fact, you could take one of these cats and shave a patch on its back and strap an ice pack to it, and you would get a royally pissed off cat that would crap in your shoes. But actually, you would also get a cat that would end up with a black patch growing in. If you shaved off some of the fur and then kept that skin cold, the fur that grew back in would grow in black. In fact, I have not seen this with my own eyes, but my wife, growing up, had a Siamese cat who was mostly an outdoor cat. And apparently, according to her, the whole cat would turn dark in winter and would lighten up in summer. And that's because we have these genes that actually put an environmentally operated switch into the cat's ability to make pigment. It'll only make pigment on parts of the body that are just a little bit cooler than the rest. In lab and in the practice problems, we mess with single genes with simple effects. The truth, what you really need to take from this, is that practically all genes show some degree of epistasis and pleiotropy, and they're also influenced by the environment that an organism lives in. So when you're thinking about human disease, some diseases we can identify as having a simple genetic basis that follows Mendelian rules. Sickle cell anemia is a great example. Tay-Sachs syndrome is another great example. Cystic fibrosis, several forms of muscular dystrophy, which is not a single disease. There's something like 50 different types of it. But for many of the types, they do have a pretty simple Mendelian basis, and they run in families. Huntington's disease has a simple Mendelian basis. But most human diseases that have a genetic component don't work that way. They're a gene, and every so often it gets reported in the media that we found the gene for breast cancer, or the gene for schizophrenia, or the gene for obesity, or the gene for heart disease, or something like that. The truth is that there are genes that can increase the chances that you'll come down with breast cancer or schizophrenia or what have you. But all of those genes are affected by epistasis. There are other genes that modify what those genes do, and those genes are also influenced by the environment that an organism lives in. You might have genes for, let's say, heart disease, but your chance of getting heart disease is also influenced by diet and exercise and probably other things in your environment as well. It's not nearly as simple as being a pea plant, where if you have, 2 copies of the big P allele, your flowers look purple, end of story, no ifs, ands, or buts. So if you don't remember anything else, remember that this, what's sometimes called beanbag genetics, where every gene creates exactly one trait, can be a useful simplification. Mendel was basically right, but for a lot of genes that we would actually want to study, the situation is much more complicated than Mendel himself ever knew, because probably all genes have some degree of epistasis that influences what effect they actually have on the phenotype. Practically all genes have more than one effect on phenotype, that's pleiotropy, and practically all genes are influenced to some degree by the environment that an organism is in. So you need to know Mendel and Mendel's laws. What you really need to know is that's not the end of the story. Okay, which raises the burning question as to what those genes actually might be. And we will pick that up at break. I will stop recording as soon as I can. I've got 9, so I will stop recording for now.